thank you everyone for your presence here tonight on a lovely hot public holiday. And before we go much further, I'd just like to pay my respects to the elders past and present of the Bunyarong, the Wurundjeri and the Wadawurrung people who are predominantly the First Nations countries groups who look after the grasslands, or on whose country the grasslands in and around Melbourne sit. I think that's probably the Wadawurrung territory goes out to where you've done your research too, I think, Alison. So it's yes, a big respect to their elders past and present, and thank you for keeping the country in such good nick until we colonial invaders came along and somewhat trashed it. So despite that, we had some good news tonight, and that is the talk by Allison Menzies on the insect pollinators of our native grasslands. Alison is doing her honours year at Latrobe University, and is we met at Vic Biocon last year. It was great to have an insect and grassland enthusiast come and speak to Grassy Plains Network there, and now we get to listen to Alison. So I will hand over to you, Alison, and you can do your thing, and that will be magnificent. I should say probably questions anytime you like, but also put them in the chat if you want, and we've got plenty of time to go through them at the end. We're not locked into 50 minutes exactly or whatever, so we'll just go by how things roll. Thank you.
Great. Thanks so much for that introduction, Adrian. Is this working? Can you see the screen? Fantastic. Okay. So hello, everyone. Yeah, I'm happy to take questions during the talk. Please do so. It helps me out. So hi, everyone. My name is Allison Menzies. I'm a mature age. I'm in the middle of my master's now at Latrobe University, and I'm studying in the Hobie lab, and I'm looking at the pollinator community in a particular Victorian grassland. So I'm here today to talk a little bit about that, but I'm also mainly here to talk about pollinators and pollination within the context of our grasslands. I've been told to presume a generalist knowledge, but I do also assume that you do know a little bit about grasslands, since I am at the Grassy Plains network. So we'll see how we go. Okay. I'd just like to acknowledge again the traditional owners of the land on which this project is taking place, and the land on which I'm talking to you from today, the Wadawurrung and Wurundjeri people respectively of the Kulin Nation, and I pay my respects to their elders past and present. This is taken at Illabarook Grassland, where I've been doing my research. It's quite a beautiful grassland, and we're very lucky to have it. It is managed by Parks Victoria. It's about 20 minutes southwest of Ballarat, a little bit past Enfield State Forest. As with all our grasslands, it is a fragmented grassland. It's set within an agricultural matrix, so it's surrounded by wheat, canola, and there is some sheep grazing surrounding it. The grassland is mostly remnant vegetation. It was apparently originally used as a long paddock, so stock did pass through it. It has actually been mined for gravel in some parts, and those parts have been re-vegged, but for the most part there is a lot of remnant vegetation there. It is a plains grassy woodland and critically endangered EPC under the EPPC Act, as most of our grasslands are. I think all of them are. It is dominated by grasses, primarily Thermida triandra, so kangaroo grass with Osterostypa and some Rhytosperma species as well. But what I want to talk about are the intertussock spaces. So the tussocks that are formed by the grasses have spaces in between, and these are what hold the floral diversity of this grassland and much of the diversity within our grasslands across Victoria. So as I hope some people have seen today, I think, did you go to a grassland earlier today, Adrian? Yeah. So hopefully today you saw daisies and lilies and maybe some heaths, probably not in flower, but geraniums, blue devils, pincushions, maybe some inch flora, and all the gorgeous annuals that make our grasslands so beautiful and special. So these create a high diversity within our grasslands, and Illabarook holds many of these types of flowering plants, which is what makes it quite special. It is closed to the public because of its diversity and because there is a lot of remnant grassland there. So I'm really lucky to be able to work within this reserve along with some other Latrobe people. So I just want to talk a little bit more about these intertussock spaces and how they work. I'm not sure how much everyone knows, so I'm just going to go ahead. So within these intertussock spaces, there is high diversity. They're often populated by forbs and annuals and flowering species, which are long-lived. These species are often thought to be often decades old or longer as individuals, and they can be considered as old growth systems. So we know that the nutrition is stored safely below ground in things like tubers, and these plants die off often during summer and they sprout new growth in spring. And they tend to reproduce underground clonally, and not much effort as a result is put into seeds because they're using all of their effort or their nutrition for storage. So seed banks in our grasslands don't last very long, unlike other ecosystems such as forests. So these flowering plants put most of their energy into storage, not into seed, and this is really important to understand when we want to understand diversity within our grasslands. So they like fire, not too much, because of the seed banks not lasting very long. Okay, so as I've said, they're fragmented, there's a low fire frequency now, and there's a low seed set, and this leads to low genetic diversity within the plants. So we're running into the risk of many populations of these plants that have very low genetic diversity because all that they tend to do is clone themselves and the seeds aren't being given a chance to grow. But there's some other factors in there as well. When we have a population, regardless of whether it's plant or animal, we need it to have a variety of genetic makeup. This allows the population to better survive disturbance events such as drought or flood, fire, disease, or invasive plants. Now, because these are fragmented grasslands, because these plants aren't reproducing very often, there's a real risk of a low genetic diversity within these plants. So what improves genetic diversity? Hope you see where I'm leading. Sorry, all right, cooperating. Okay, pollination. So specifically cross-pollination. So sexual reproduction as opposed to clonal reproduction is what gives us diversity for our species. And so pollinators deliver this crucially important service to our plants. And, you know, we depend on plants to live in many, many ways. So cross-pollination has been shown to improve seed sets for native species. A study in 2000 of milkmaids, Spicardia umbellata, has found that there were higher rates of viable seeds after cross-pollination. Plants can actually self-pollinate, so one individual can actually produce its own seeds, but they tend to have low viability. They're not as healthy as if there's been cross-pollination. So we know that this cross-pollination is really important and it helps promote diversity within our plants. But this aspect of our vegetative health is It's of low interest in some ways to managers of our remnant grasslands, and much of our remnant vegetation, full stop. I'm not here to denigrate anything to do with people who manage our grasslands, because they do an incredible job with the few resources that they have. It's just that this aspect is not perhaps thought of as often as other ways to manage our grasslands. All right, so just a quick summary about pollinators. Globally, 87% of flowering plants require pollinators. This is a global figure. Australia falls somewhere around here as well, but we don't know for certain. Pollinators in turn use floral resources for nutrition, for habitat and reproduction. Pollinators perform cross-pollination for flowering plants, which drives genetic diversity, and it provides resilience against disturbance. So now I want to discuss a little bit more about what a pollinator is. So when we are studying pollination ecology, it's really important to understand what a pollination event is. So we define pollination as the transfer of pollen from the anthers of one individual to the receptive stigmas of another individual of the same species. And fertilization does need to take place. So this is what we technically consider a pollination event. So not all visitors to our flowers actually perform this pollination event. So what we need to understand is that we have what we call flower visitors or floral visitors, and some of them are pollinators. So some species are there, they're doing this various things. They might be hunting, they might be eating, they might just be hanging out, but not all floral visitors are pollinators. And then what leads on from that is that some pollinators are more efficient than others. So the most efficient pollinator is, you guessed it, probably bees. They're really good at pollinating. They have lots of hairs, they collect lots of pollen, and they move it around from flower to flower really well. Some other species perform pollination kind of incidentally. So in my study that I'm performing, there are what I've been calling SBBs or small black beetles. They are around by the billions, I think, in our grasslands, and I'm sure you've all seen them on flowers. For these, they're tiny, they're so small, and a grain of pollen for these is probably about the size of maybe even a tennis ball or a bit smaller to them. So they only carry a couple of grains of pollen on each individual that I'm seeing at the moment. So whilst they don't carry a lot of pollen, there are so many that they're probably moving pollen around in our systems just through sheer force of numbers. So we would call this kind of pollination more incidental. They're not really specifically pollinators as such, but they are doing some pollination work. So I'm just trying to set this up so that we know that we've got floral visitors, and then some of them are pollinators, and some of them do a lot of pollination, some of them don't do too much. There's another aspect to pollinators that I just want to talk about as well, which are generalists and specialists. So we have kind of like a spectrum. So we have generalists here and we have specialists here, and you know, a lot of insects, and I'm mainly talking about insects today. A lot of insects fall along this kind of spectrum. So we have generalists on one end. So these are species, bees are good at this. They visit many species across many families. They're not that discriminatory in who they visit. So they pollinate lots of different species. They keep lots of different plant species alive. And the bees are our best generalists. And we also, conversely, I guess, have specialist flowers as well, or specialist plants. Daisies are quite a good example of this. So this is a patch of everlastings that I visited at Mount Arapiles about 2 years ago, and I got to sit for about an hour with my camera. So what we can see here is this one kind of patch of flowers we're attracting. There's probably about 13 different species altogether here. So some of them, can you see my cursor? Yeah. So here, it's not the greatest photo, but this is a spider. This probably isn't doing any pollination. It just sits here all day. Sorry about the quality of that photo. And underneath this, we've got an assassin bug, which again tends to sit on the outsides of flowers. They're not touching much of the pollen, and they're there to hunt and eat. The rest of these species are probably doing more pollination than these two. But this is a really good example. Daisies are great generalist flowers in that they'll attract a lot of species because they're nice and open and you can land on them really easily and you can get to the pollen quite easily if you're a pollinator or if you're after some pollen. So we've got these generalists, which can be a pollinator species or they can be a plant species. And having these within our communities creates a lot of resilience. So what I mean by that is that, for instance, within a pollination and plant community, if one of the plant species happens to drop out because of disease or something like that, the pollinators that are generalists aren't going to be particularly put out by that. They'll still be able to fly around, they'll still be able to visit lots of species. And so this community can sort of withstand a lot of disturbance. So we've got generalists here, and on the other end, we have specialists. So specialists, so some pollinators only pollinate one or two plant species specifically, and some plant species depend on only one or a few pollinators. And the best example of this specialization is orchids and wasps, and we have some really good examples in Australia. So some wasps are the exclusive pollinators of a single orchid species, and I'll show you in a minute. This is actually a video. Some orchids depend solely on a single wasp species to reproduce, but often while these wasps don't need the orchid itself to reproduce, often these orchids, as I just said, need the wasps to maintain a population. So this is a video that I'm about to show you. It's a slow motion video of a Calladenia. I think this is a tensa. It could be a tentaculata. I am not certain because I keep getting them confused. My fellow master's researcher, Andrew Bird, who is working with Ryan Murphy, is investigating tensa and its pollinators at the moment. And this video sort of shows how this orchid species has evolved to be physically attractive to a single wasp species. There's only one wasp species that is attracted to tensa. Tensa emits pheromones that attract, that mimic female wasp pheromones. And so these wasps will come here, they'll get right in, they're going to try and mate with this orchid, and then the orchid taps it on the head and it deposits some pollen on the wasp's head, which we'll see right now, I hope. Oh no, sorry, try again. So I was getting right in there, trying to mate, and that orchid is about to take full advantage of that. And you'll see the wasp fly off with a load of pollen deposited on it.
Fabulous shot.
Yeah, it's a fantastic. Andrew's got some amazing videos. But yeah, so that orchid has deposited all of its pollen on that one wasp. These orchids really do put all of their pollen in one basket and depend on this one individual to go and take its pollen away to another orchid. So this is a real specialist relationship. And what I think Andrew is seeing at the moment around Victoria is that we are finding, sorry, I've gone with him on a couple of trips. So we're finding that there are populations of wasps that are tensa pollinators, but there are no tensas around. So these wasp populations, they don't need the tensa to actually reproduce or live, but where wasp populations are not, you're not going to find these tensas. If a wasp population dies off for some reason, that tensa population will last only as long as the individuals that are currently there. So they are extremely dependent on the wasps to reproduce. Just checking the chat, sorry. All right, so this is specialists. So we have generalists that are doing lots, and then we have these specialists. And so what these specialist relationships do is maintain the real diversity within our floral communities. So they keep the rarer species going within our communities. And so these are the relationships that are at risk, most probably at risk from any sort of disturbance like disease or invasive species. So just understanding these aspects of pollination helps us understand what's happening with our pollinators and what they're doing in our communities. Well, all right, so I'm just going to quickly go over our key invertebrate pollinator orders, and this is the situation in Australia and worldwide. So we have Hymenoptera, which consist of our bees, wasps, and ants. Ants don't do too much pollination as such. They don't have much hair on their bodies. They're quite small. I've watched countless hours of videos of ants just crawling around a flower head, not coming into contact with anthers or stigmas at all. But bees and wasps do a lot of pollination relatively. Coleoptera, which are beetles, they have made up the bulk of my study at the moment. About a third of the invertebrates I've been capturing is Coleoptera or beetles. We've also got Diptera, which consists of sciapids, Bombylidae, and other flies. Flies do a lot more pollination than I ever dreamed of before I started studying pollinators. And flies are amazing as pollinators. They're some of the cutest things you're ever going to see. Hemiptera, so bugs, true bugs, do some pollination perhaps. And we've got Lepidoptera, so our butterflies and our moths. So globally, these are our key invertebrate pollinator orders. Now, in Australia, there are unfortunately few studies of our pollinator communities. There are studies of bees or bees and butterflies or beetles only, but we really lack kind of the bigger invertebrate picture. So it's a real shame though, because a lot of the studies that we have show us some really interesting things about what we know about pollination. So a study in 2005 of visitors to a flowering jarrah canopy, so Eucalyptus marginata in Perth, Western Australia, noted that, so there's a study in 2005 looking at a jarrah canopy. Previous to this, only 15 species had been recorded as visitors to jarrah, and it was presumed that avian pollinators, so birds, were the primary pollinators. And the study that this 2005 study found at least 83 species from 39 families were actually pollinating the canopy, and most of the primary pollinators were Hymenoptera. So it's really changed how they would look at jarrah and who's pollinating it and the community around it. So, and also at the moment, there is a huge study in Queensland in the Atherton Tables by Mark Hall, who's, I don't know if you've heard of him, does a lot of pollinator studies. He's now based in Bendigo, but they're doing a big community assessment up there to sort of tackle as well this sort of dearth of pollinator studies, of pollinator community studies. So the studies that we do have show us a lot about our pollinators. So how can we better understand our pollinator communities when we don't have much information on them? We just need to keep surveying them, which kind of leads me to a little bit about my research on pollinator communities. I won't go into this too long. So my study is trying to understand how a new survey technology, which is called eDNA metabarcoding, looking at how effective it is at surveying pollinators in a grassland compared with traditional survey methods. And then I wanted to use the combined survey data I got from all this to better understand the local floral visitor and pollinator community. So I'm just quickly going to explain eDNA metabarcoding, which is really briefly, I don't want to go into this too much. So we and everything, whenever we touch anything, we leave trace DNA on it. So invertebrates, when they're on a flower head, they leave their trace DNA, and we call it environmental DNA or eDNA that's left on these flowers by the invertebrates. So I've been collecting these flower heads and I process them to collect that DNA that's been left on them. and we get that DNA out and we get sequences, which we call barcodes, and we match those against sequences that are in databases. And that then tells us who's been on our flowers in a nutshell, very simply. So we call this meta barcoding because I'm dealing with hundreds of barcodes all at once that are being pulled off these flower heads. So we wanted to have a look. It's a relatively new technology. It hasn't really been used that much yet on flowers, and we wanted to see how it compared with traditional methods. So just quickly, the advantages of meta barcoding are that it can capture up to 5 days worth of eDNA. It captures day and night visitors, which a lot of survey methods don't, and it can capture hopefully rare and smaller species that we can't capture with a lot of methods. The disadvantages is that it is only as good as our databases, and here in Australia we don't have that much data on our invertebrates, so we're kind of battling that. And it can't distinguish between a pollinator and a visitor, so it can't tell me if that what's been on the flower has been taking pollen off it or not. It can just tell me that it's been on that flower. Pardon me. So the traditional methods that I've been comparing it with are sweep nets, vane traps. So vane traps are probably about this big. They're like big blue yogurt. They're like big yellow yogurt containers, and they've got like this stuff that comes out the top, and the bees can go in, and they get trapped in there. Well, not bees, inverted flying insects. Also camera traps. So we set up cameras just to watch flowers. That's what I spent my Easter doing. Don't know about you, watching videos of pollinators. And observation. So we've had people just sit and watch flowers for 20 minutes at a time, record down who they've been seeing. And so all of these methods, they capture a brief time period for the most part. The vane traps can be left out overnight, provided that there's no rain. They're mostly confined to daylight, and they need a taxonomist to tell us who we're looking at or who we've captured. And again, for the most part, they can't distinguish between visitors and pollinators. So this is just kind of the study that I'm doing. So the species that I've been surveying, I picked some really common species across the grasslands. So I think you're going to recognize all of these. I didn't want to run into too many problems with using anything that was particularly rare. And I also really wanted to capture a diversity of flower head types to see if I could get a really good picture of the community depending on these flower heads. So we've got grey parrot pea, milkmaids, chocolate lilies, common everlasting, and possibly my favourite, the blue pincushion, which I really love. So each of these was surveyed across 2 days period using the sort of traditional methods, and at the end of each 2-day period, I collected 10 flower heads from each, and we've used that to process for the DNA stuff. All right, so just a quick summary of what we caught. So there were 925 invertebrates captured. We made 467 observations, and for the meta barcoding, there were 265 confirmations of presence of individuals. Don't worry about too much about this ASV stuff. It's not, it confirmed the presence of individuals 265 times, essentially. So just a quick, some sort of visual summary. This dark blue is the DNA meta barcoding and how many individuals roughly it identified. And this is just to sort of look at it as a comparison. So the sweep nets here are in yellow. They did a lot of hard work. Caught a lot of insects with these sweep nets. The pale blue is the vane traps. Observation really didn't do much for the most part. And orange is the camera traps, which identified a lot more than I thought they would. But this is just sort of to give you an idea of what we were dealing with at the end of the first part of this study. So what we wanted to understand is how much return we were getting for effort. So the sweep nets gave us 737 individuals, which I'm still sorting through. And while that's a really good number, and it took 190 hours worth of sort of field work and lab work to get these identified, to order for insects, and it was 3.88 sort of identifications per hour of effort. But the eDNA, for 57 hours of effort, we got 4.65 identifications per hour of effort. So it's a really good method of identifying. And eDNA also got us down to probably family or subfamily for the most part. For all of these, we are still working on getting them down to hopefully subfamily. And that's going to require a lot more effort. So just that is kind of my study. The first part of my study is to sort of understand how this was all working. So what I'm doing at the moment is we're looking at all of the insects that we caught and trying to get them identification sort of down to subfamily as well. What we did identify is acari and mites technically aren't invertebrates or insects in a way, but they were caught, and spiders, technically, they can be considered a little bit apart from the pollinators. But we got cockroaches, springtails, beetles, millipedes, flies and bugs, and ants, bees and wasps, butterflies and moths, mantis. We got one praying mantis. We got some, I think we got one damselfly, which was a real shame. I was a bit heartbroken about that because it's a couple of grasshoppers and katydids and a lot of thrips. So this is what we captured. So the orders that I mentioned earlier are firmly in the mix here. So we can be confident that we did get a really good idea, like really good picture of who's in our grasslands and who's pollinating them. No, wrong way. Okay, one moment. All right, I won't go there yet. So this is what we caught at the moment. These are floral, what I consider to be all floral visitors. We are still in the process of pulling pollen off individuals. Probably about 300 had pollen on them. So out of about nearly 900 captured individuals, only about a third had pollen. So this is going to really pull down what we consider to be our picture of pollinators versus the floral visitor picture that I have at the moment. And it's still at a really high level, which is order. So we're going to try and pull all these invertebrates down a little bit more detailed. But what I did is, with this data that I had, knowing who everyone visited, I created a bipartite plot. It's a really neat way of understanding a pollinator or floral visitor community. So it's got all these lines. Don't get too, look, I'm going to break it down a little bit, but it really shows well, the thicker the line, the more visits that are going on from that particular order. And don't worry too much about reading the orders at the top. I will go into a little bit more detail. So look, not that much going on, but what we can see is that Coleoptera, which are the beetles, are pollinating, they're visiting, sorry, all of these species that I surveyed, and in large numbers because they've got the thickest lines. Diptera, which are flies, are also doing the same, and they're also doing it in large numbers as well. Hymenoptera, which are the wasps and bees, are again visiting all of the flowers and doing it in large numbers. And we've also got bugs. Spiders are there as well, visiting all of the flowers, but spiders, we know, just don't do much pollination. So I'm going to sort of break this down. I'm going to partition it, and we're going to talk about these invertebrates in a little bit more detail now. All right, so I'm going to cover Hymenoptera first. So these are our ants, bees, and wasps. So they visited all of the species I surveyed. 48% were wasps, 20% are native bees, which was wonderful. Even better was that Apis mellifera, so honeybees, were only 1%. We only saw 2 Apis mellifera in the study, which was just so good not to see them. Like, it was wonderful not to have them in the mix, just to see that they're not around in this grassland. And ants were 31%. Many of the wasps here are probably parasitic or parasitoids and too small for effective pollination. But we'll have a look at some of them. I've got a pollinator visitor right now, sorry. So let's look at bees first because they're the best. All right, so over here, we've got an Apis mellifera, which is a European honeybee, which was brushing. It had so much pollen on it. It's such a good pollinator. It was brushing pollen off onto one of my sheets of paper because it had too much. And that's all that we'll say about that. So let's have a look at these. So most of the bee species shown here are what we call solitary species. One of the photos may not seem like it, this one, but in Victoria, we mostly have solitary bees, which means that they don't form collective hives like honeybees do, or what we see in warmer climates. The females tend to nest underground, sometimes in groups, but the males will sleep either by themselves or collectively, like we can see here with the megachills or the leafcutter bees. This was amazing to see. This wasn't at Illabarook for my study. I'm just showing, like, this is more of a selection of different species that we've got. This was up in Wodonga. So what we did find is some halictids, possibly exoneura, which are possibly reed bees. These are really good examples of a bee that can really get into pea flowers. I don't know how well you can see it here, but this is the same little bee. He's gotten right into the pea flower and he's dived right in, and he's exposed the anthers here, which then rub straight onto its belly where it can collect that pollen. So they are really good at pollinating our peas, some of these little bees. Honeybees tend to be too big to get in there to do that. So this is one example of what our native bees can do. This megachile or a leafcutter bee is just the cutest little thing. It's got like little red hair up here. I kind of think of it as like a punk bee. It's also a long-tongued bee, which just means that they have longer tongues than most. You can see a good example down here. I know this is blurry, but it's at the point is to see this really long tongue of this blue-banded bee here. So we can get right in and get to the pollen. So these are leafcutter bees. Sometimes you might see them in your garden only by the round little holes they cut into your leaves on your plants. We've got lasioglossums. I think these are lasioglossums. A lot of these identifications here are kind of preliminary at the moment. I've only just started working on beetles, and so they're a bit more confirmed than a lot of these other ones. But lasioglossums do a lot of the hard work in Australia for pollinating. You see them everywhere on everything most of the time. And you can see these two have got quite a lot of pollen on them. And lasioglossums also do what's called sonication or buzz pollination. So what they do is probably, so hopefully this works. I actually did this for some educational purposes, but if we pretend, so this is a representation of a, oh my God, I'm blanking, a Dianella species, all right? And so when we see the Dianella, all the pollen is kept in here. It's not on the outside of these anthers, all right? It's held in here, and we can see it with that Amygdala as well down here. On here, like the pollen actually isn't on the outside, it's held in here. And so what sonication does is we've got specific bees in Australia. Hopefully, can you see this? Our little blue-banded bee. So what this does is with its jaws, it holds onto the top of these anthers. It buzzes at a frequency, and that buzzing, do it here, shoots the pollen out, and it lands on the bee. On the bee's abdomen here. So it has to vibrate at a specific frequency for the pollen to come out of most of our lilies. I hope that worked. I hope you could see that. So honeybees can sort of do it. They're not very good at it, but our bees are particularly good at it. Blue-banded bees and the sweat bees can do it. when I was surveying, there were so many chocolate lilies, there were so many of these little lasioglossums, and I was holding on to a chocolate lily at the bottom trying to take a photo, but I could actually feel the vibration on this lily as the fly was buzzing to get the pollen out. Like you could just feel these occasional buzzes on the plant while it was doing its buzz pollination, which was extremely awesome. So that's one type of pollination as well that I don't know how many people know about that, but it's something as well that our bees perform. So the masked bee over here that I haven't really talked about is kind of an oddity in Australia in terms of pollination. As you can see, it doesn't really have any hairs on it. It's a masked bee. They are really, really tiny. So they are floral visitors only. They visit flowers to eat pollen and drink nectar, but they don't actually move pollen around. So when we find them in sort of pollinator studies, they do tend to be excluded from the studies because we know that they don't carry pollen on them. So these are definitely bees. Might look like a wasp. It's a form of protection, but they don't actually do much pollination. These are some examples of the bees that we have. All right, so just looking at wasps. For the most part, the wasps that we found are parasitoids. So they are tiny, tiny wasps. Most of these are around 2 millimeters long, and we can only sort of see them under the microscope. They're hovering around. They're looking for something to lay eggs in, for the most part. All right, whether or not it's a plant or whether it's not, it's another species that's also on a flower head. But we did find the blue flower wasps, which I'm sure you've seen buzzing around many of our grasslands. And also another thinid species as well was captured, and these were captured on the Burkhardias, so the milkmaids. Milkmaids are, I think the milkmaids had quite a specific pollinator set, so we found quite a lot of flies and a lot of flower wasps, some bees, but a lot of flies on these, I think because they're white and because they're pink, and they attract flies because of that fleshy pink color on them. Not too sure about the wasps, but yeah, that is something to look into further. But that was really interesting. So these are some of the wasps that we found. Wasps come in many shapes and sizes and colors, and they're gorgeous little things, but they don't tend to do too much pollination unless they're a dedicated kind of flower wasp. All right, so Coleoptera were on all the flowers a lot of the time. So Coleoptera are beetles. Nearly 70% of the Coleoptera that we captured are these small black beetles, which are around 1.5 millimeters. Again, these are really only visible under a microscope, but these are the things that you can see all the time on flowers. And we also found quite a lot of these Philoticus species, which for the most part were found in vane traps, and actually for the most part were on the Hypercharis, the invasive daisies. So I'm not too sure. Once we do sort of pollen analysis for these, we'll see what they were doing. Oh, and when I say pollen analysis, yeah, we're taking pollen off the flowers and we are going to analyse who they've been visiting as well. And these are some of the beetles we found. So these are the tiny little small black beetles, which we see on many flowers. They particularly seem to love, these are on the wiry buttons, but they were all over the Chrysocephalum apiculatum, so the common everlastings. We saw a lot of these nectar scarabs. This is in a similar sort of subfamily to the nectar scarabs, and that has to be a pollinator. Look at all that hair on it. It definitely captured a lot of pollen, and we pulled quite a lot of pollen off that one. These are just kind of interest. We've found a leaf rolling weevil, which is only about 2 millimeters long. That's probably not moving any pollen around because it's got nothing to collect pollen. We found lady beetles, and we've also found a snout and bark beetle, which is closely related in a way to the weevils. But these are just some of the beetles that we're finding. We've collected about 300 beetles. I think that we're going to end up probably with only about 30 of them actually being separate species. Diptera flies. So again, they were on all of our species in varying kind of numbers. A lot of small to medium flies, which are either 5 millimeters or less in size. We had some syrphids, which are our pollinators flies. And then we had a lot of other flies like blowflies, or just kind of your common medium-sized flies, I guess we call them. Flies are extremely hard to identify down to species or even genus. I'm leaving them to last. Like I could in them all, just should. So let's talk about flies and pollinators. They're often overlooked. Their flies are extremely robust. They do a lot of pollination in regions that other insects might find a little bit too inclement to live in. So studies up in the Alps have found that flies are probably more common than bees, particularly when it's windier or cooler. So they don't mind that kind of weather, so they'll be out and about doing a lot of pollination. These are these are all flies, all right? So these are little bee flies, which are just the cutest things. They really like peas. We saw them definitely over the peas quite a few times. And this is another little bee fly as well. You can hear these, you can hear them if they're nearby. They've got a higher tone than other kind of flies. Like if they're nearby and you hear something that's a little bit higher that's buzzing around, it's probably going to be a bee fly or a hoverfly. Well, also these villas, this picture is actually taken, this picture is taken in Castlemaine of this particular species. I haven't seen it in the grassland that I've been in, but this again is a fly. So their flies are frequently misidentified as either bees or wasps because they don't really have any defenses. So they use mimicry as a form of defense. So this is why we're seeing all of this diversity within these flies. And these are all dedicated pollinators. So hoverflies, which I'm sure we've all seen, very common. And these drone flies, and this is also another drone fly. So these are all related as well as these ones too. And then we've got a huge collection of kind of what at the moment I'm calling misc, Diptera, which are just many flies that we will be identifying at some point. So I think that flies, like these ones are doing a lot of pollination. We just don't really see it because we think that they're others. We think that they're either bees or we think that they're wasps. Particularly this one really flies around like a bee, like a honeybee, and it does tend to look like a honeybee if you're not closely looking at it. And so the Diptera, Coleoptera and Hymenoptera are the ones that I'm going into in the most detail. So this is kind of the other floral visitors that we have kind of come across. A lot of spiders, but again, they're not doing much pollination, but they're definitely there in large numbers visiting the flowers. We've got a couple of Orthoptera. This is obviously a juvenile, which makes it very hard to identify. Amongst the Hemiptera, we've had a few mirrored bugs, and I am about 90% sure this is what's called a sundew bug. So these are specifically associated with Drosera. I have spoken with a person who is studying them, and they are specifically associated with sundew plants. And we saw a hawk moth, which for me was a highlight as I'd never seen one before. So this, I'm pretty sure, was out because of the Burkhardia or the milkmaids. Forester moths, which I suspect everyone has seen, and we found quite a few skipper butterflies as well. So these are definitely pollinating. This we think might be pollinating. It was found in a vane trap overnight. And these probably not so much pollinating, but definitely all floral visitors. So also what I did was I plotted the DNA results on top of the bipartite plot, just because I wanted to see how much of a broad picture it was giving me compared to everyone else. And it is giving me a pretty good sweep. So it gives us good value for money. It also gives us a nice community that is similar to the one that these other methods are giving us. So eDNA is looking pretty good. And I just wanted to sort of say, in terms of the community that we found in Illabarook, the Hymenoptera, the wasps and bees and the bugs, the flies and the beetles are quite generalist species because we're finding them on all these different types of flowers. So they provide a lot of resilience within this invertebrate community, but deeper identifications and pollination, pollen analysis do need to be done to pull this down to a pollinator community. And there's that. And so what I'm going to be doing is we're going to be identifying all the captured invertebrates. I'm also going to get their DNA and upload them to databases as well, so that we've got these in our databases. And we're currently pulling pollen off individuals. I'm creating a pollen library, and I'm going to match the pollen that's on the insects to the plant pollen of the species that I've been surveying, and this will give us a pollinator community. And just really quickly, this is what pollen looks like under a high performance microscope. So interestingly, the Burkhardia and the Arthropodium kind of look a little bit similar because they're both lilies. What's really interesting is that the common everlasting, which is a daisy, has very spiky pollen, which is part of what we think makes Asteraceae such a successful species. It really sticks to its pollinators, to its insects, because of all these little spikes. And this is the blue pincushion, and these are the Dilwenia. So just, they're kind of cool. And so I've got a lot of people to thank, not least of all Susan Hovey and Nick Murphy at Latrobe University for my research and my interest in invertebrates. So yeah, thank you very much for your time and attention. And I will take questions if you've got them. Yeah.
Magnificent. Thank you, Alison. That was eye-opening. It was fabulous. Okay.
Oh, good. I'm not an expert, but yeah.
Sounds pretty expert, and I'm sure you're just going to get more and more expert as you go along. There was an awful lot of work you're going to do for your masters. It's
yeah, I know.
yeah, so okay, bit of overachieving never goes astray either. Chris put a couple of questions in the chat. Did you want to speak them out, Chris, instead, or shall I just read them?
Yeah, just to say about, I mean, I've been amazed reading more about moths and the number of species and the number of plants that are pollinated by moths. And I wondered how much of that was happening in grasslands. And you mentioned with the eDNA side of things, you can look at more at what's happening at night. So I wondered too, I suppose, if you chose particular plants that might attract moths, whether they're scented or more white flowered or whatever, maybe you get different results.
I did, yeah, I did.
Lepidoptera or, yeah.
I didn't particularly think about that. I am glad I did the milkmaids because they're white. So yeah. That hawk moth, we're pretty sure it was there for the milkmaids. But again, one of the problems is that unless you're out there doing like sweep netting at night, or which is very hard to do because then you can't see what you're capturing, or unless you're doing you know, the sheets with the lights. Sorry, I'm blanking, but I know the Entomology Society of Victoria has been doing that for the last couple of nights up in Bendigo. So unless you're doing that, we don't actually know who our night pollinators are. We just don't have that data. So the eDNA, whilst it might pick them up, it can't necessarily tell me because the databases aren't giving me that data. Does that, like, eDNA is only ever as good as the databases that hold the information.
Yeah.
So I think it's good to have it. It's good to have the eDNA data, and hopefully as those databases get more and more added to it, as we rerun that eDNA data through them, it'll come up with stronger hits on who these pollinators are. At the moment, I'm getting kind of weak.
Yeah.
It's like to family. Does that make sense?
So is the actual design of a lot of moths good for picking up pollen? Is that
For the most part, it is. So for the most part, moths, hawk moths are really specific pollinators. So
Yeah.
put them there. Moths, for the most part, yeah, because they're like butterflies in that they like to be able to sit on top. Just think about how they sit when they're at rest. They want to be able to just sit and drink straight from something. Hawk moths, we can kind of think of it in a similar way to birds as pollinators. They hover and they kind of, their heads are like this and they'll drink from something. So we are pretty sure that hawk moths pollinate the native Nicotiana species that we have, the native tobacco, because it's got
long tubes.
A long tube. Hawk moths have long tongues and they hover. Nicotiana overseas is pollinated by hummingbirds. So hawk moths kind of perform a similar pollination service here to that. So they pollinate a different way just because of how they sit. But for the most part, other moths are similar to butterflies in that they probably do like to just sit on top of something that's easy to sit on.
Dip in.
Yeah.
Fabulous talk. Thank you.
Oh, thanks.
Yeah, Alison, all of these species that you're identifying as pollinators, apart from the honeybee, are they all native species? Like,
yes.
Yeah.
For the most part, there are some beetles that are pests, agricultural pests. I mean, there are some invasives there, but for the most part, they are all native species.
Okay. And is there a risk that honeybees are taking away resources from our native pollinators? Or
definitely.
Yeah, definitely. Okay.
Definitely a risk. They're extremely efficient pollinators. They collect pollen like no one else's business. They do take resources.
Okay. Someone asked how long the eDNA can last before it becomes denatured. Yes.
Yeah. So, okay. So out in the open, eDNA can last. Studies have found that it can last for up to 5 days in good weather. One of the problems with this study was that I was surveying last spring. So trying to get days of just even sunshine was a problem. So in between all of these days, there was a lot of rain. So I'm actually resurveying at the moment. I'm heading to Illabarook tomorrow briefly to do some more collection of flower heads. And hopefully I'm going to get stronger results this year. But eDNA, I mean, in terms of denaturing sort of in general, once you've collected it, if you store it correctly and keep it cold, it can last for a very long time. It can possibly last for years, but
okay. Alan asks if you've seen any March flies. He has one on a daisy in his garden.
I don't think I have out there, Alan.
Yeah, that's interesting. We think Roger Farrow had a look at this and thought it was a male, and that there's some logic in that because it's the females that prefer the blood.
Yeah, we haven't had any like bothering us. Definitely not. I'm not sure when they come out. I don't know too much about Diptera. I might have avoided them, but I don't think we have come across them, Alan.
Okay. Well, Parks Victoria, Tillis is looking after this reserve. I know you're trying not to. You're going to be very diplomatic, but how well are they looking after this particular reserve?
They're doing the very best they can. There have been burns. They have ripped warrens. The reserve has quite a lot of Acacia paradoxa, and my supervisor as well is about to have some more students start looking at paradoxa across Victoria and its diversity and looking at origins of paradoxa because it behaves as a weed in some species, and in Illabarook it's harbouring rabbits. So they're trying, Parks Vic have done work in trying to eliminate those, but funding just keeps getting pulled. So there's only so much that they can do. So
Okay.
I've told them where I've seen 1 or 2 patches of things like Patterson's curse, so that they can deal with that quickly.
Okay.
I think they are
Yeah.
hoping to do a burn soon as well, in a year or 2.
Deb Reynolds has a question.
Hi, Ellie. Sorry I'm a bit late to this, but extremely interesting. I spent ages looking at what was pollinating Pimelia, and it was flies.
Oh, wow.
Exactly what you're telling me, flies.
Yeah.
And there's 1,000 species of flies.
Yeah.
And I took many, 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 many pictures.
Mhm.
And so I have heaps of stuff. That illustration is stunning. Who did that?
I did the illustrations.
Yeah,
because
on your
I
thank
had
you and questions.
deadlines.
Sorry?
I had deadlines, so I did everything except the work I was meant to do.
Oh, that's beautiful. We need that on a t-shirt or something. It's gorgeous, but not with thank you and questions, but with the other stuff. But I have lots and lots of pictures, and I take pictures religiously of beetles and all sorts of things, hoverflies and wasps. And yeah, I saw a honeybee once only on Pimelia spinescens. Ever,
Oh, wow. And
ever.
where
And
were you surveying a particular area, studying?
well, I know I did it all over the state, remember? I went to all my PhD sites, because we did every single site, and we found, again, spiders, because spiders are predatory for the flies. I have photos of the spiders catching the flies. And I couldn't see, it's amazing. I have some amazing pictures. So I'm really curious, how you're doing it and what you're doing, because this is very interesting stuff, but mainly flies, because there's so many flies. And this is during winter, remember? Pimelia flowers over winter. So
again, we see a lot of flies because everyone else doesn't like it below 17 degrees.
Well, what I would find is I did exactly what I went out in the middle of the night. I went out all sorts of times, and I just found nothing was happening at night much because it's freezing. But after about sort of 10, 11 o'clock, warmed up a bit, and suddenly the flies were active. There were flies everywhere. If you walked up to a plant, you'd have probably 10 or 20 flies move off because you're upsetting them. So that was when they were doing their thing in the middle of the day, because
when we were
it was winter.
surveying the milkmaids, the flies were just everywhere. I really like them.
Yes. So I'm extremely interested in these insects and all the little pictures, because I've got lots and lots of pictures. So I'd love to catch up with you and have a chat.
Yeah, definitely.
I think we should.
Ellie, too, if I could just add that from the Australian Plant Society, we'd love to have you come and speak.
Thanks.
And also we are potential source of funds as well. So if you need a grant for some of this work. All right, I'll send you my contact details. Cheers.
Thanks, Chris. Appreciate it.
Good job. Okay, so you've got a job out of this and a grant already, and it's like, and it's only 7:30. Doing very well. Is there any correlation between well, there is, but I don't know what it is, between the flower colour and the types of visitors to the flowers?
There is, to an extent. So we know that different species see different along the colour spectrum. They see at different frequencies. So we know that bees really like purple and blue because that's what they can see. And we know that butterflies do quite like yellow. Sorry, I do. I'm trying to remember. I actually was teaching this a couple of weeks ago, so I really should know it. But what I can say is that we know that different insects see different frequencies, and so they are definitely attracted to different colours, and plants do take advantage of that. So we can see that in like these yellow daisies, the chrysophyllums were really interesting in this study because they had the most diversity, they had the most species identified in terms of what I physically captured and also in what the eDNA captured. And until I kind of went back and looked at some of my field data about each day of surveying, I surveyed those probably around November at this last year, and I was like, yeah, it must have been warmer, like more species out. They really like it. And it actually turned out that when I surveyed the chrysophyllum, it was actually the coldest days I'd surveyed in. It was sitting around 17 degrees. And that kind of, I think, goes to show just how popular daisies actually are, in that even on these cold days, they still just had so many visitors to them, and they're flat. The nectar's easily available. If you're an insect, it's just easy to land on it, you know, and just move around. So,
well, yellow's a predominant colour too. You know, Podolepis in the picture behind me. You see bees all over those, and they're like having bee parties. There's hundreds of them on. It's hilarious. So yellow generally is a lot of our grassland species, and murnongs and rudidosus and Grassocephalum. So heaps of them are yellow. And also the other thing I find very interesting is we need to do more sequential studies, because each flower has its time. And then there's another flower, and then there's another flower, and then there's another flower, and each flower, that would be very interesting. Pimelia is another one that, you know, flowers during winter and then has sequential flowering from, you know, during winter it's spinescens, then it's glauca, then it's curviflora. So we have a sequential flowering for Pimelia, which is a white flower with.
I do love a good curviflora.
Yeah, they have beautiful perfume, and they get moths in the evening too. So I have some really good photos of all the moths on certain moths, not many, mostly flies yet again. Sorry.
But yeah, we do know that colour and also flower shape are related to who's visiting them. And it is a relationship to some extent.
And temperature too.
And temperature. So, I mean, the hard life of a pollinator is that you can only really survey at 17 degrees and above, and it can't be raining. So life is tough to me.
What's the other thing? How far does pollen go? Because I only ever found studies that showed that pollen could last for maybe 100, you know, 100 feet, 100 meters, maybe, possibly on a pollinator.
I honestly don't know.
No, well, that's the thing, is how far can it go between, you know, a grassland is continuous, and where does it stop and start, and how far could you actually mix? Because our grasslands would have extended for 100 kilometres, but actually, if you've got fragmented grasslands, how far can a pollinator take it?
Honeybees carry stuff for like 10 kilometres, don't they? They forage like a really long distance from their hive. So wouldn't that be the case for other species too?
It can. It depends on so for bees, particularly honeybees, because they've got a base that they need to keep coming back to, they actually don't go that far because they've got to return to that hive. But for species such as our solitary bees, which, you know, they'll sleep wherever they feel like, although the megachilds that I had a photo of that were in that huge group actually kept returning to that same area each night to sleep. But they're not bound as much by having to return to a hive. They just kind of move around. So depending, I guess, on the circumstances of the particular insect, some of them will travel a lot further. I imagine flies as well move around quite a bit because they don't have too many nesting concerns either. But for some species that do have a hive, they don't travel as far, but the other ones can kind of just move wherever they want in a way. So they might be traveling a lot farther. Kind of answers that, I think.
Brilliant. More questions. Lots of people out there still who are being silent. It's not right. Is there sorry?
I was going to say, I can see there's a comment from Paula at Wyndham Wildlife Gardens who's got Nicotiana flaviolens. Please check out what is looking at them, Paula, because we don't actually have much data on if the hawk moths are actually, we don't, because I've done a thing with ALA sort of superimposing hawk moth data on top of the Nicotiana flaviolens to see if there's some sort of co-occurrence, and we don't actually have enough data to say for sure. So it would be really good for you to have a look one night, set up a white sheet, put a lamp on it, and see who's visiting around the Nicotiana. That would be awesome. Cool.
Fantastic. All right. Well, if that's it, we might wind up unless anyone's got a last pressing question. No. Okay.
We have your email, Ali, so that we could contact you.
Yeah, sure. I'll put it into actually. I'm putting it in the chat now.
Thank you.
And this will be up on recorded and on the Grassy Plains website in due course. It might take a week or so for us to get our act together, but we will get there. It's indeed. So huge thank you to Allison for this deluge of knowledge about these things. And yeah, we look forward to hearing what further results from your studies, and yeah.
So do I.
More. Yes.
Thank you very much. Thanks so much for asking.
Thank you very much.